Good afternoon, everybody. My name is Kate Fitzgerald. I'm a professor at UMass Chan Medical School here on the East Coast in the greater Boston area, and it's a real pleasure to welcome you to another Global IMMUNOTALK. Today we have a fantastic speaker, Russell Vance, who's professor and a HHMI investigator at UC Berkeley. And I'm going to briefly introduce Russell in a moment. I just want to remind everybody to be sure to sign in for next week's speaker, October 5th, and that will be Franca Ronchesi. So for today's talk, we have Russell Vance, and as I said, Russell is at UC Berkeley. I'm just going to give a brief intro to Russell. So Russell was born in the US, but actually raised in Canada. He did his BSc and a master's at Queen's University in Ontario in Canada, and then moved to UC Berkeley for his PhD in immunology, and then was a postdoc, postdoc actually, both with John Michelanis and then I think later with Bill Dietrich in microbiology and then the genetics department at Harvard Medical School. And then Russell has been at UC Berkeley since starting his independent program in 2006. From there, he's really risen through the ranks to professor in the Division of Immunology and Pathogenesis. And he became a HHMI investigator in 2013. He's received numerous awards, so I'll just mention a few here. So as a graduate student, he received the Harold Weintraub Award, which is a really impressive, nationally recognized award for a PhD thesis. Not surprisingly, he's gone on to great things. So he was a Damon Runyon Cancer Research postdoc who's also had a HHMI predoc fellowship. He's had, for example, the Burrs Wellcome Fund Path Award, a Cancer Research Institute Investigator Award. He currently has an NIH Merit Award and has given many lectures and received numerous awards for his work. So Russell is very famous in the innate immunity field, really known for his work on inflammasome biology, particularly the NAPE NLRC4 inflammasome, his work on STING activation. He was the first to demonstrate that STING is a receptor for cyclic dinucleotides. And then he's gone on to really sort of focus on effector-driven immune responses and studied in detail a number of different bacterial pathogens. So Legionella, Pneumophila, Shigella, and I think what we're going to hear about today is some of his more recent work in the area of TB. So Russell is going to talk about tuberculosis as an unconventional interferonopathy. And then before we get started, Russell, we usually have our speakers, this is particularly geared towards sort of trainees and other junior faculty. Can you share a little bit about an impactful decision that you made in your career and how you think that shaped your wonderful success to date?
Yeah, thanks, Kate. And thanks for inviting me to give this global IMMUNOTALK. I think that you and the other organizers have done a great service to the immunology community by organizing this series. It's really been incredible, and I'm glad that you're still continuing it.
Yeah.
When I think back on my career, I would say the most impactful sort of decision that I made pretty early on was to really, I made a very conscious decision to get training in microbiology. So my PhD was in immunology. I was in David Raleigh's lab for my PhD, and it was pure immunology. I was studying natural killer cell receptors. Yeah. And at the end of my PhD, I kind of felt like that the purpose of the immune system was to defend against pathogens, and yet I hadn't touched a pathogen the entire time of my PhD. And I felt like I didn't even understand pathogens. They were like kind of foreign creatures to me, and I felt like if I want to understand the immune response, I need to understand pathogens because that's what's driving the evolution of the immune system. So I made this decision to kind of leave immunology behind, and I went to John Michelanis' lab, which is a pure micro lab. And, I have to say things didn't go that well. It was a big transition. And, I don't think I was terribly successful as a postdoc, actually, in my first postdoc. But in retrospect, looking back, what I learned during that time was, how bacteria are, like the biology of the microbiology and the biology of microbes. And that's something that's carried through the rest of my career. So, I ended up doing a second postdoc because the first one wasn't as productive as maybe it should have been. But, actually what I learned during that time was incredibly valuable. So I would just say I encourage all immunologists to, embrace microbes and really don't treat them like a bag of pamps, really try to understand their biology, because I think that's, understanding microbes is the key to understanding the immune response.
Yeah, no, and I think your work, I think the power in understanding the microbiology as well as the immune response to the pathogen. And, pathogens teach us a lot, right, about how the immune system is activated and evaded. I think we learned some of the most important things by studying how pathogens manipulate these systems. Yeah. Great. All right. Well, thank you, Russell. We're looking forward to your talk. If you can share your slides, and then at the end, I'll come back on and we can remind people how to ask questions through the Global IMMUNOTALK and through Twitter. Great. It's all yours.
Okay. So can you see the slides?
I can see everything. Yeah, looks good.
Okay, great. Well, great. Thanks, Kate. And again, it's a real pleasure to be able to give this talk. Tuberculosis, as Kate kind of alluded to, is a pretty new area for my lab. I wouldn't really even consider myself a tuberculosis expert at all, but it's something that I think is really important, and we're trying to put a big investment into trying to understanding this very important disease. So let me begin with the most important slide, which is, of course, to acknowledge people in the lab who have done the work. And in particular, most of the unpublished data that I'm going to be showing you today comes from a really terrific postdoc in the lab, Dmitry Kotov. And he has been assisted by an undergraduate in the lab, Ophelia Lee, and I'll show you some of her data. Dmitry and Ophelia have been building on work that was done previously in the lab by a great graduate student, Daisy G, and a current graduate student, Kristen Witt. And if you're working on tuberculosis, it's always a team enterprise. And so other people in the lab that are working on TB who really, help out with everything to do with working on this difficult pathogen, Marion and Stefan. And as I said, you can't really work on TB in isolation. You need really great collaborators and colleagues, especially if you're new to the field like I am. And I would say that, our work on TB would have never started if it hadn't been for a sabbatical visit from close colleague Heron Darwin, who was a TB researcher in NYU. And it was really her visit that spurred us to actually take on this important problem. I'll mention that a collaborator that we've worked with closely, Igor Kramnik, has been also very critical. And at Berkeley, I have two terrific colleagues who also work on tuberculosis, Sarah Stanley and Jeff Cox. We share the biosafety level 3 lab, and really, I think we couldn't do anything without their help. So with that, the pathogen that I'm going to be talking about today is this bacterium. It's an unusual bacterium, Mycobacterium tuberculosis, and it was discovered 140 years ago by this gentleman, Robert Koch. And this bacterium, as I already alluded to, is extremely difficult to work on. It has to be handled in a biosafety 3 laboratory. It's very slow growing. It takes something like 2 to 3 weeks to form a colony on an agar plate. But at the same time, M. tuberculosis is also, I think, without doubt, the worst infectious disease killer of humanity. It's responsible for something like a total of 1 billion deaths. And even today still kills about 1.5 million people per year. So it's a real serious problem. Prior to Koch's work, interestingly, tuberculosis was widely believed to be an inherited disease. But it was really, the careful work of Robert Koch that established that it was actually caused by this pathogen, Mycobacterium tuberculosis. This is actually Koch's 1882 drawing of the tuberculosis bacilli in the lung. This is a more modern image, actually, from my lab. The TB bacteria here are shown in pink, growing in the lungs of a mouse. And I think what was so brilliant about Koch's work was that he was able to establish a methodology that we now call Koch's postulates to rigorously prove that the bacilli that were observed in the lungs of these animals were in fact the cause of the disease and not just passengers in the lesions. And this is one of his quotations that I particularly love. As he wrote, his experiments allowed him to conclude that the bacilli present in the tuberculosis lesions do not only accompany tuberculosis, but rather cause it. These bacilli are the true agents of tuberculosis. And I do really love this quote because, as I will discuss, the challenge of distinguishing cause and effect remains a major issue in TB research, and the two are often conflated. So it's a real challenge to distinguish cause and effect. And despite the fact that we've known about this bacterium for 140 years, the pathogenesis of tuberculosis remains very poorly understood. What we do know is that it's aerosol transmitted, and it has a very low infectious dose, perhaps as low as 1 CFU. And it's transmitted through the aerosol. So when you breathe in the bacteria into your lungs, the first cells that seem to be infected are the alveolar macrophages. And I think basically this part is kind of well established, but then after the bacteria actually infect these macrophages and start to replicate is where our understanding starts to get a little bit more confused. So it's clear that there's some kind of initial innate response to the bacteria. You have influx of immune cells like neutrophils and monocytes. And the bacteria will grow initially during that innate phase, probably kind of uncontrolled. And then the adaptive immune response kicks in, and there are basically a whole series of very heterogeneous and complex outcomes. So in some people, it appears that the bacterial growth is actually contained within these structures that form called granulomas, and the bacteria there's kind of a standoff with the host, where the bacteria are prevented from growing out of control, but they're also not eliminated, and so they reside in the lung perhaps for many, many years. In some other people, the immune response may actually be successful in eliminating the bacteria. And then in a minority of people, actually probably something like 10%, the bacteria are not contained or eliminated. Instead, they kind of replicate out of control. Okay, so there's this wide sort of series of sort of heterogeneous outcomes that can occur, and it remains actually extremely poorly understood why the response would tip to one outcome or another. So active disease is associated with this uncontrolled bacterial growth. So if you contain the infection, usually you don't show any symptoms at all. But if the bacteria start to grow out of control, it can form very large lesions in the lung, and then the patient can start to show symptoms. They're going to cough up the bacteria and transmit it to the next host. And this state here, this disease state, is the one that we're trying to understand. It's associated with a lot of bacterial replication. It's also associated with a lot of inflammation. And again, what's really hard to disentangle here is what's causing what. Is it the bacteria that are causing the inflammation, or the inflammation somehow allowing the bacteria to grow? And so a major challenge is to distinguish cause and effect. So this is the question that we're interested in and that we're trying to address, which is, in the subset of cases where infection does progress to this active disease state, why does the immune system fail to control TB? And, there've been many answers provided in the field to try to answer this question. And some of the standard explanations that you'll read in literature are that MTB has mechanisms for hiding or blocking the immune response. So the idea here is that it's immune evasive or immune suppressive in some way. It's hiding in these granulomas, for example, where maybe interferon gamma, which is a very important cytokine for controlling TB, maybe interferon gamma can't reach the infected cells or control the infection. So that's kind of one general class of explanation. Another explanation that you'll also see in the literature is that MTB can exploit inflammation, so that inflammatory response that occurs is actually benefiting the bacteria in some way. Maybe it's allowing the bacteria to obtain nutrients. Maybe it's creating tissue damage, which provides a niche for the bacteria to replicate. So this is another explanation that you'll hear in the literature. And I think what's interesting is if you consider these two possible explanations, you can kind of see why the field is in kind of maybe a state of confusion. Because in some ways, these are almost exactly the opposite of each other. I mean, on one hand, we're saying TB is blocking inflammation and that's how it grows. And in the other case, we're saying it's enhancing inflammation and that's how it grows. So which is it? And it's quite confusing to reconcile these two different kinds of explanations. So I think we don't really understand actually how this all works. And so, my lab's been very interested in trying to understand this better. And the alternative, and I would say non-mutually exclusive explanation that I'm going to focus on today is, so these things, may be true to differing degrees in different situations. But the idea that I want to focus on today is this sort of third idea, which is that tuberculosis is able to replicate well in people because of its ability to induce a type 1 interferon response. And the consequence of this interferon response is that it actually suppresses the effective immune responses that are necessary to control TB. So what I'm proposing is that TB is kind of inducing this kind of immunosuppressive state. And so basically what I think is so confusing about this, and what you have to kind of try to get your mind around, is that when you look in the lung of a patient with tuberculosis, what you see is a lot of inflammation. And so it looks like there's too much inflammation. But what I'm proposing is that what looks like excessive inflammation might actually be concealing a state of immunosuppression. And I think this is kind of an unfamiliar idea for many people, that how could inflammation kind of hide itself? How can inflammation be hiding kind of a state of immunosuppression? But actually, in other fields, like in cancer, for example, I think this is kind of an idea that is kind of more widely accepted. So I'm proposing maybe that there's some similarities actually between the immune response to TB and the immune response that we see like in other chronic inflammatory states like cancer. Okay, so this idea that type 1 interferons play an important role in tuberculosis is not new. Early work from Anna Guerra, for example, this paper published in 2010, she compared the transcriptional response in the blood of patients with active TB compared to patients with latent TB. And what she observed was that she could see transcriptional induction of many type 1 interferon-induced genes. And so clearly there's this interferon signature that occurs during tuberculosis. But I have to admit that when this paper came out, I wasn't really sure what to make of it, because again, it wasn't clear to me whether this interferon signature was actually what was responsible for the disease, or maybe it was just a consequence, right? I mean, these patients with active tuberculosis have a lot of bacteria replicating in their lungs, and so maybe it's not at all surprising that they would have an interferon response. And so again, there's this challenge of distinguishing cause and effect. However, this interferon signature has been reproduced in many other studies. And some of these studies, this is one that I kind of particularly like because it's a cohort study where they follow a group of patients who have latent disease and then watch them as they eventually convert to active disease. And they ask, what kinds of signatures in the blood predict the progression to active disease. And what they found actually was that a type 1 interferon response is really one of the best predictors of progression to active disease, and it can predict active disease 12 to 18 months in advance. So this kind of suggests that maybe there's a certain chronology here, that you have that interferon response first, and then you develop active disease, which kind of is maybe suggestive of a causal relationship. But of course, it is still just a correlation. It's actually not really showing any causation here. So ideally, what you'd want to be able to do then is establish a causal relationship between the type 1 interferons and TB by, for example, say deleting the type 1 interferon receptor from mice and asking what effect that has on TB disease. But a problem with this kind of simple idea is that the standard C57 Black 6 mouse that everyone uses really only weakly induces a type 1 interferon response after TB infection. And this is a now classic paper, actually, from my colleague Sarah Stanley and Jeff Cox, 2007, when they were looking at this interferon response to TB, and they showed that it required an important virulence factor of TB. And in fact, in the lungs, they could see induction of an interferon response in Black 6 mice. But this is a very modest response. And when they actually did the experiment I suggested, which is to delete the interferon receptor, it didn't seem to make any difference to the growth of the bacteria in the lung. So there wasn't any functional role for type 1 interferons in TB control in the lungs. They did actually see a difference in the spleen, but not in the lungs. Okay, so So it's very difficult if we believe that in humans, the interferon response is somehow driving disease. It's very difficult to dissect that mechanistically without some kind of mouse model. And so when I started working on TB, we started with the idea that maybe we should try to find a better animal model. And we became very interested in this work that was published from Igor Kramnik's group, showing that C3H mice, actually a different strain of mice, are highly susceptible to tuberculosis infection. And what Igor really in heroic work was able to show was that this susceptibility maps to a genetic locus called what he called the SST1 locus, or super susceptibility to tuberculosis locus. And so C3H mice carry the susceptible allele of this locus, and the B6 mice carry the resistant allele. They are SST1R. And if you make congenic mice, so B6 mice that are B6 all throughout their genome, except for this one locus where they carry the susceptible allele from C3H mice, you can see that these congenic mice harbor much higher burdens of TB in their lungs and have much more severe disease. And so this is the heightened inflammatory response that I said accompanies active TB disease. You can see there's lots of consolidation of the lung here, lots of inflammation, lots of bacteria. And at the time, it was proposed that it's this excessive inflammatory response that's actually driving disease. But again, what I'm going to propose to you is that what looks like excessive inflammation in these lungs is actually what's actually happening underneath the response is actually a state of immune suppression that's allowing the bacteria to replicate. Okay, so we were very interested in these SST1S mice, and so Daisy, a graduate student in the lab, was curious whether or not type 1 interferons might be driving the susceptibility of these mice. And so we got these mice from Igor. He very generously shared them with us. And what Daisy did was to try to treat these mice with antibodies that block the type 1 interferon receptor, so anti-IFNAR antibodies. And what she saw was that actually these anti-IFNAR antibodies dramatically reduced the CFU burdens in these mice, something like 100-fold. Daisy also crossed the SST1S mice to IFNAR knockout mice, and again got the same result. So this actually shows now a causal relationship. The type 1 interferons seem to be responsible for this excessive bacterial replication. And when you get rid of the type 1 interferons, that basically restores the mice to the B6 phenotype, which is the resistant state. Very interestingly, if you look at IFNAR knockout mutations on the B6 background, there's really no phenotype here. And again, this is because the interferon response in the B6 mice is very, very modest. And we only have a strong interferon response in these SST1S mice. And so that's where the interferons actually have a functional role. So I'm not going to talk about this in great detail, but one of the things we were interested in was, what is the gene within this SST1S locus that's responsible for this susceptibility phenotype. And Igor's lab had originally proposed that there's this gene called IPR1 that's responsible. It maps to this genetic locus. Unfortunately, because this locus is incredibly repetitive, it was extremely difficult at the time to actually make IPR1 knockout mice. But with the advent of CRISPR, we're actually able to make those knockout mice. So IPR1, its other name is Sp110. And Daisy was able to make Sp110 knockouts. And very surprisingly to us, we found that actually these mice were not susceptible to TB. So it seems like IPR1 and Sp110 are not the gene that is responsible for the susceptibility of these SST1S mice. It turns out that actually this gene IPR1 or Sp110 is actually part of a family of transcriptional or chromatin regulators. So it has genes involved in recognizing chromatin marks. And it's actually part of a small family of related proteins, which includes this very interesting protein AIR that may be familiar to many immunologists. But these genes all cluster together on the chromosome. And actually, these three, Sp110, Sp100, and Sp140, are all encoded within this SST1 locus. And very interestingly, in C3H mice, Daisy found that this gene Sp140 was also defective. So it was another candidate gene to explain the phenotype of these mice. And so then Daisy went on to make the Sp140 knockouts, and with help from Kristen, was able to show that these Sp140 knockouts are actually highly susceptible to TB infection, and phenocopy the SST1S mice. So now we believe that it's the causative gene within this locus is actually Sp140. Having B6 mice that are deficient in just a single gene is a very useful reagent. It allows us to make crosses to other knockouts, for example, all on the B6 background. And so basically now everything else I'm going to show you in this talk is using these Sp140 knockouts as this model for susceptibility. Okay. As I already kind of alluded, the phenotype of these mice is that they make an excessive type 1 interferon response after TB infection. So this is looking at interferon beta 1 levels, transcript levels in the lung after TB infection. You can see that SST1S mice make more interferon beta compared to B6 mice, which have a very low interferon response, right? That's shown here. And this heightened interferon response is phenocopied in the Sp140 knockouts. And we can also see induction in these mice of interferon stimulated genes. So one that has become particularly interesting to us is this ISG called IL-1RN. That's the name of the gene, but the protein product that it encodes is IL-1 receptor antagonist. And the reason why we were particularly interested in this interferon-induced gene is because we know that IL-1 is a really critical factor for controlling TB replication. And so it was very interesting to us that interferons were inducing this antagonist that actually blocks IL-1 signaling. And you can see that it's really strongly induced in the Sp140 knockouts as compared to B6 mice. Okay, and so what we've now gone on to do, and this is now work from Dimitri and Daisy Kristen, is to cross our Sp140 knockouts to IFNAR knockouts and to IL-1 receptor antagonist knockouts. And you can see here, just like with the SST1S mice, if you cross the Sp140 knockouts to IFNAR, this rescues the susceptibility of the mice. So again, proving that type 1 interferons are not just being induced as a consequence of the infection, they're actually causing the susceptibility. Okay, so that's an important point. And very interestingly, similarly, if we knock out the IL-1 receptor antagonist gene that I mentioned, this also rescues the mice. And in fact, I think this is one of the more remarkable results that I've ever had in my lab, actually, is that eliminating just one copy of IL-1 receptor antagonist, so the heterozygous mice, are largely rescued, okay? And this is shocking because type 1 interferons induce hundreds or thousands of different genes, all of which could be playing some role during TB infection. But it seems like eliminating just one copy of one of these interferon-induced genes is actually sufficient to partially rescue the mice. And if you eliminate both copies, the bacterial burdens in the lungs are driven right back down to what we see basically in an Sp140 IFNAR double knockout. Okay, so based on these results, what I want to propose is that tuberculosis might actually be what I'm going to call an unconventional interferonopathy. So we typically think of type 1 interferons as really critical for inducing antiviral responses. However, type 1 interferons are not that helpful during a bacterial infection, such as with TB. If you have excessive type 1 interferons, this can cause, this is widely known to cause a series of autoinflammatory diseases. The classic example is this disease called Aicardi-Goutieres gene, where you have excessive type 1 interferon production, and this is very pathological. It's a very severe autoinflammatory disease. But what I'm proposing is that during TB, what the type 1 interferons are doing is not causing excessive inflammation, but so it's not really a conventional interferonopathy, but it's rather an unconventional interferonopathy, because what the type 1 interferons are doing here that's causing pathology is actually being immunosuppressive. So effectively, what we're proposing is that they're inducing proteins that actually block the effective immunity. Okay, so type 1 interferons are hindering responses by interfering, for example, with IL-1 signaling, and also I will propose interferon gamma signaling. Both of these cytokines, IL-1, interferon gamma, are critical for control of TB, and type 1 interferons are known to negatively regulate these responses. So these responses are critical for controlling TB, and type 1 interferon is interfering with that. So for example, probably through many mechanisms, but one that I've shown you already is through induction of this IL-1 receptor antagonist. Okay. I should just mention at this point that, this idea that interferons are detrimental during tuberculosis is not particularly new. I already mentioned Anna Guerra's work. And by the way, I think Anna Guerra is giving one of these talks in a few weeks, so I encourage you to listen to her talk as well. But many other people in the field have also contributed to this idea. There's some early work from Robert Maudlin's work studying Mycobacterium leprae, so the causative agents of leprosy, so not tuberculosis, but still a related mycobacterial infection, shows that type 1 interferon actually suppresses interferon gamma triggered responses in humans. So this seems to be possibly sort of a common theme among different mycobacteria. This very important paper from Kat Meyer-Barber suggested very interesting mechanisms. So not IL-1 receptor antagonists, but other mechanisms involving lipid mediators that might also be interfering with IL-1 signaling. There's some recent paper from Anna Guerra, and I think this is kind of an interesting report showing perhaps the relevance in humans, which is that people carrying mutations in IFNAR seem to be resistant to tuberculosis. Okay, and this is just really a small number of the papers that together really, I think, contribute to this idea that type 1 interferons are really playing this pathological role during tuberculosis. What hasn't been so clear is what they're doing that's bad, but I think there are hints that it might be through this immunosuppressive mechanism that I'm mentioning. Okay, so here's where we are. This is sort of our working model, and that what we're proposing is that this transcriptional regulator Sp140 is really critical for repressing the production of type 1 interferons. So when you are an Sp140 deficient mouse, for example, you no longer negatively regulate type 1 interferons appropriately. And when you get this TB infection, you induce these type 1 interferons. These induce immunosuppressive factors like IL-1 receptor antagonists that block the effective immune responses that, for example, IL-1 mediated responses. Okay, so what I want to talk about in the last part of my talk is further work that we've gone on to try to really flesh out some of the details of this model to actually understand what's really happening. And in particular, we're interested in what is the source of this type 1 interferon, what cells are actually producing it, and then what cells are responding to that interferon. So I'm going to touch on both of these. To begin to address the first question, which is which cells are making this interferon, Dimitri in the lab has done a series of single-cell RNA-seq experiments, and the results are shown here. So he's compared B6 mice to Sp140 knockout mice. He's looking specifically at myeloid cells in this experiment, and indicated in the blue dots are the cells where we actually see interferon beta transcripts. And when we got these results, I would say that we were somewhat disappointed because in B6 mice, for example, I think we could find 6 cells in total that were producing type 1 interferon. In the Sp140 knockouts, which make a stronger interferon response, I think we could see something like 25 cells or something like that, but it was still very few cells. And this actually seems to be kind of a general problem with studies of type 1 interferon. It's actually hard to find cells that actually make type 1 interferon. I think this is because interferon is produced in these very short bursts. So they make it and then they stop making it. And so at any one moment, if you capture a series of cells, it's hard to find many cells making interferon, even though potentially many have made it or will continue to make it. So the single cell RNA-seq actually wasn't that useful to really nail down which cells are making interferon. And because the idea was that they're making small bursts of interferon, we thought that maybe another approach to really track these cells might be useful. And so we took advantage of these interferon beta reporter mice that Dan Stetson has made. These are actually not yet published. He shared them with us very generously prior to publication. And basically what these mice are knock-in mice that have an iris TD tomato downstream of the interferon beta gene, and then very importantly also encode a CRE. The TD tomato part, we found it very hard to find very many TD, like it was hard to find interferon beta producing cells in the lung. It was also hard to find TD tomato producing cells in the lung. But this CRE allowed us to adopt kind of a lineage tracing strategy to identify the cells that maybe made interferon in the past. So basically, Dimitri crossed these mice to our Sp140 knockouts and also crossed them to this AI6 reporter, which is basically a fluorescent green protein downstream of a floxed stop cassette. So the idea here is when a cell produces interferon beta, it'll produce Cre, it'll excise the stop cassette, and then the cell will be permanently marked. Even if the interferon beta transcript disappears, the cell will be permanently marked with this very bright green fluorescent protein. Okay, so this actually works quite nicely. This is an experiment Dimitri did in looking in the lungs of TB-infected mice at different cell populations, and he's infected the mice with a M-cherry fluorescent TB. So we can track which cells are infected and then which cells are expressing this AI6 reporter. And T cells kind of serve as a negative control here. We don't expect them to be infected with TB. We don't expect them to make much interferon. Indeed, we don't see much expression of the AI6 reporter in these cells. The main cells that are infected in the lung after TB infection are actually interstitial macrophages. We can see a lot of these cells are infected, but interestingly, of only a very small minority of the infected cells actually seem to be turning on this reporter. And most of the cells that have turned on the reporter are actually not infected with TB. So this might be because these cells have cleared the infection, but I think that's actually quite unlikely because it's very difficult actually for macrophages to kill TB. And so what we think actually is that what might be more likely is these actually might be bystander cells. So many of the cells that are producing interferon might not themselves have ever been infected, but might just be bystanders. I also want to point out that we, in the lungs, we can actually see also that there are PDCs that are present. These are known producers of really abundant producers of type 1 interferon. There's very few of these cells in the lung, but on a per cell basis, they produce a lot of type 1 interferons. So we actually, as I'll discuss in a minute, I think these might actually be very important contributors to the interferon production in TB-infected mice. They're not infected by TB, but again, they might be acting as bystanders that are responding to ligands in the environment to produce type 1 interferon. Okay, so basically one of the surprises here was that most of the interferon-producing cells in the lungs seem to be not themselves infected, but actually bystanders. And we were able to kind of validate this observation by actually looking using immunofluorescence in the lungs of these mice. So in B6 mice or Sp140 knockout mice, there's much more bacterial reps. So bacteria are shown here in pink. There's many more bacteria in the Sp140 knockouts. The AI6 cells, is the interferon reporter positive cells, are in green. And what you can see both in B6 and in Sp140 is that many of these green cells are actually not themselves infected with bacteria. They seem to be proximal to regions that are heavily infected, but they themselves might just be bystanders that are producing a lot of this interferon. Okay. The PDCs, though, were the cells that actually really caught our interest. And this was for a variety of reasons. First, it's well known that they are extremely abundant producers of type 1 interferon, but there's also data from the literature. This is a paper from Shabana Khatter's group that says that, basically studying non-human primates, doing a very extensive characterization, that her group found that 3 defining features of pulmonary TB in macaque lungs include the influx of plasmacytoid dendritic cells and interferon responsive macrophage population and activated T cell responses. So obviously, it's nice to see in this model, which is a closer model to human TB, that there seems to be this influx of PDCs. However, of course, in the primate model, it's impossible to actually do that cause and effect type of experiment to determine whether these are actually important during the infection. And so this is the experiment that actually Dimitri took on using our mouse model. These are Sp140 knockout mice that he crossed to mice in which you can relatively specifically delete these PDCs. These are PDC DTR mice. And so when you add DT to these mice, you can see that the number of PDCs is decreased. And importantly, if you look in the lungs at the levels of bacteria in the lungs, you can see that depletion of these DCs reduces the CFU burden in the lungs. So suggesting that these are actually these PDCs are actually contributing to the interferon that promotes the bacterial replication. And this only occurs on mice that are Sp140 knockout, where we have that heightened interferon response. In mice that are Sp140 functional, they have one copy that's sufficient to restore their resistance to the infection. Depletion of PDCs has no effect. So, and that makes sense because these mice are not overproducing the interferon. Okay. So bottom line is that we think that these PDCs are an important source of the interferon that's driving pathology. Of course, in humans, it's extremely difficult to really get a handle on this, but we were able to collaborate with a group of David J. and Emery, where they had actually looked in human tuberculosis patients. These are culture-confirmed patients. And what they were able to do is stain for this PDC marker, CD303, which is also called BDCA2, which is a pretty specific marker for PDCs. And very interestingly, they could see PDCs localizing again to the periphery of these granulomas. So this is where the bacteria will be replicating, and these would be the bystander cells around the edge. Very suggestive that they may be also playing a role during human tuberculosis. Okay, so one of the big questions is, if these PDCs themselves aren't infected with the bacteria, how are they getting activated during TB infection? What we know about PDCs is that they're usually activated by sensing extracellular nucleic acids. And they're activated by recognizing those nucleic acids through toll-like receptors like toll-like receptor 7 or toll-like receptor 9. So the question is, what is the source of these extracellular nucleic acids? There's lots of dead and dying cells in these TB lesions, so it makes sense that there might be a lot of DNA ligands to activate these PDCs. And one very intriguing idea, which has been suggested by Anna Guerra's group, is that maybe a source of DNA that might be very important is neutrophil extracellular traps or nets. And so I presume many of you may be familiar with this, but when neutrophils undergo a particular kind of cell death, they can actually spew out their DNA, their genomic DNA. And the idea is that this DNA forms a kind of net that traps and kills the bacteria, but this DNA could also serve as a ligand to activate PDCs. And we can actually identify this DNA because this DNA has a very specific histone mark, a citrullonated histone H3. Okay, and I'll get back to that in a minute. One reason to think that neutrophils might be playing a very important role here is that if you look in the lungs of these Sp140 highly susceptible mice, they're really basically full of neutrophils. There's tons of neutrophils in these mice, as you can see here. And very interestingly, if you cross these mice and rescue their susceptibility by crossing them to the IFNAR knockouts, you can see that the number of neutrophils is actually decreased. So basically what I think is going on here is that maybe there's some kind of positive feedback loop where if you produce a lot of type 1 interferons and suppress the immune response to TB, then the bacteria will replicate out of control, and this will stimulate the recruitment of lots of neutrophils. But the neutrophils themselves might also be a source of these nets or DNA ligands that are actually responsible for inducing type 1 interferons, and you end up with this kind of positive feedback loop, which you can interrupt by deleting the IFNAR receptor. And I'll show you in a second, you can also interrupt it by depleting neutrophils. This is just further kind of suggestive images that perhaps these nets are actually playing an important role. I mean, I don't think we've proven that by any stretch, but it is very interesting when you look in the lungs of these infected mice that, again, you can see here we're staining for these citrullonated histone H3, which is a marker for nets. And you can see really abundant nets in the lungs of these Sp140 knockout mice, really absent from the B6 mice. And again, these nets seem to be co-localizing with these interferon-inducing cells, at least in this image. And this is the kind of thing that we're now going on to try to quantify in more detail. Okay, so it's just a suggestive image that maybe indeed these might be playing an important role. And these were images collected by undergraduate in the lab of Ophelia. Okay, so as I already promised, to kind of close the loop here, you can do an experiment where you deplete neutrophils, and you can see that this actually does, similar to IFNAR mutation, you can interrupt that feedback loop by depleting neutrophils. And so basically in Sp140 knockouts, they have high bacterial burdens. And if you deplete neutrophils, this is using the 1A8 antibody, Ly6G, that specifically depletes neutrophils, you can largely rescue these mice. So I also want to point out at this point that in addition to forming nets, neutrophils are also the main producers of IL-1 receptor antagonist. So if we put this all together, then it leads to this kind of model where there's this dysregulated type 1 interferon production that somehow suppresses immunity to TB. You get out of control TB growth. You end up with lots of neutrophils. And then you have these positive feedback loops that promote TB pathogenesis. So one thing that we're proposing is that these neutrophils are producing these nets, which are stimulating PDCs to produce more type 1 interferons. And then you get this kind of positive feedback loop in this way. The neutrophils that are abundant in the lungs are also producing a lot of this immunosuppressive protein IL-1 receptor antagonist that blocks IL-1 signaling, and IL-1 is critical for control of TB. So when you repress this signaling, now you have more replication of TB, more interferons, and you get another positive feedback loop. So we think that maybe this is sort of basically how tuberculosis spins out of control in that subset of patients. Okay, so in the last few minutes here, I'm just going to talk a little bit about some of our work about trying to address this question, which is, there's a bunch of arrows here. What's actually happening? What cells are actually the main ones that are responding to this type 1 interferon, and how is that leading to loss of bacterial control? So we can return to our single cell RNA-seq experiments and look on what cells, in the TB lung are actually expressing the type 1 interferon receptor. Again, this wasn't terribly helpful because it turns out that most cells produce the type 1 interferon receptor. This just shows that actually, we can detect gene expression in single cell RNA-seq, but it's not helpful because basically all the cell populations we're looking at express this interferon receptor, and they have the potential to respond to interferon. But what we would like to know is actually which cells are responding to type 1 interferon. And so to address this, we collaborated with Brian Bryson's group at MIT. He had had done some experiments to try to look at, develop signatures basically of cells that are responding to interferon beta compared to other stimuli. And in particular, one of the challenges here is that the genes that are induced by type 1 interferon and type 2 interferon or interferon gamma are actually very overlapping. However, there are some unique genes that are preferentially induced by beta or preferentially induced by gamma. And this allows us to develop a signature of a cell that is receiving an interferon gamma stimulus or receiving an interferon beta stimulus or both. And so we map these signatures onto the single cell RNA-seq data set. And what we can see here is that in B6 mice, which are resistant to TB infection, we see a strong signature of response to interferon gamma. So, and in particular, the interstitial macrophages, which are the main cells infected by TB, basically most of these cells show a signature of responding to interferon gamma. And so this should be able to restrict the replication of the bacteria. In the Sp140 knockouts, which have this heightened interferon response, we see a lot more blue and green, right? And that makes sense because there's a lot more type 1 interferons here. We see in particular a very strong interferon response by the neutrophils. As I'll show you in a minute, we don't actually think this is contributing much to the disease outcomes. But very interestingly to us, we see in the interstitial macrophages that there is a population of macrophages that seem to be responding to interferon beta and not responding to interferon gamma. So these macrophages we would predict would be very permissive to bacterial replication because they wouldn't be inducing the genes downstream of interferon gamma that are important for bacterial control. Okay, so we suspect that maybe these macrophages are the permissive niche where the bacteria can replicate out of control. And we wanted to try to address, on which cells is interferon actually signaling by crossing IFNAR floxed mice to different CREs that specifically delete the interferon receptor, either in neutrophils or in all myeloid cells. So this is the data where we delete IFNAR from neutrophils specifically using MRPH CRE. And what we can see here is that these mice are still more or less susceptible to the infection. So they're not, unlike an IFNAR Sp140 double knockout, which are protected, these are still significantly susceptible, and they more or less resemble the Sp140 knockout littermates. So deleting IFNAR from neutrophils doesn't seem to rescue the mice. By contrast, deleting IFNAR from all sort of myeloid cells using lysam CRE, this does actually seem to largely rescue the mice. So this would be suggestive, if it's not neutrophils, that maybe it's macrophages or monocytes that are the key interferon responsive cells that actually leads to permissiveness. Okay, so I'll conclude here with our sort of working model, which is that when you become infected with TB, there's this induction of type 1 interferons. This might be a low level of interferon, but it might be sufficient to impair the ability of macrophages to control TB replication. That will lead to more TB growth, and this will induce the recruitment of neutrophils to the lung. The neutrophils are, in this case, we think, playing a very important immunosuppressive role. They're producing factors that suppress the effective immunity to TB. So one thing that we think that they're doing is producing these nets that stimulate PDCs to produce more type 1 interferons, and I showed you deleting PDCs can help interrupt this feedback loop and lead to protection. In addition, neutrophils are a major source of this IL-1 receptor antagonist. And I showed you deleting IL-1 receptor antagonists can also rescue the mice. And this is probably through the IL-1 receptor antagonist is blocking IL-1 responses, which somehow, and it remains to be understood exactly how that works, help promote control of TB as well. Okay, so and of course, this transcriptional regulator that I still think we're very interested in understanding how it works, but it seems to play an important role in repressing the type 1 interferon response. And so when you don't have this, then these feedback loops get out of control. But if you do have this transcriptional regulator, then that suppresses these feedback loops and helps contribute to resistance. Okay, so then I think I will end. I think I acknowledge all the people that contribute to the work, but I'll just emphasize again that a lot of this data is unpublished, and it's work from Dmitry Kotov, who is going to be on the job market. So if you're looking for a fantastic scientist who's really working on a very important problem, I encourage you to get in touch with Dmitry. And so with that, I will stop sharing and pass it back to you, Kate, I think.
Yeah, that was...
We don't take questions, right? It's all going to be on Twitter.
No, unfortunately, it'll all be on Twitter because I have lots of questions. There's just a lot of, yeah, just both at every step of that little cascade that you outlined, there's a lot of really interesting questions. Fantastic, absolutely fantastic. Right, so just to remind everybody, yeah, we do questions on Twitter. You know, you can go right now or at your leisure. Russell is an active tweeter, so he'll keep an eye on his Twitter account and respond when he gets a chance. But he'll take questions directly at Russell E. Vance, and also use the hashtag #GlobalImmuno. And thank you all for joining. Thanks again, Russell, for a terrific talk, and we'll see you October 5th for Franca Ronchesi. Great, good to see you, Russell. Thank you.
Yeah, nice to see you, Kate. Okay, take care.